Welcome to Genomics Unlocked, where genomic science and webinars converge. Today's episode focuses on immune repertoire profiling with DNBSEQ.
Right. Hello, everyone. I think the time is now 2 p.m. In Singapore time. So we have a 2-hour session. So I believe we can start now, and hopefully more people can join as well. So welcome to the Genomics Unlocked, where genomic science and webinars converge. We're using this GoToWebinar platform for today's session. Please feel free to interact with us through the polls and Q&A features available for all attendees. Your insights and queries enrich our discussion and bridge the gap between this curiosity and understanding. Our platform also features an on-demand sections where you can catch up on all our previous webinars, ensuring you staying updated on the cutting-edge advancement in genomics. I am your host, John. I'm the MGI Senior Business Development Manager for Asia Pacific. We're dedicated to bringing the forefront of genomics research and discussions to both professionals and enthusiasts alike. So in the webinar series today, which the title is Immune Repertoire Profiling with DNBSEQ platform, we have the honor to present to you 5 speakers from our 4 industrial partners. These are from Takara Bio, Singleron, Merexis, and MGI Research, respectively. We're excited to present a panel of these guest speakers who will share their insights, and each speaker will bring a unique perspective from their field of expertise in their specific assays for immune profiling, utilizing genomics in bulk, single cell, and spatial field. So a short intro of immune repertoire profiling. We're exploring immune profiling's crucial role in disease research and oncology, delving into B and T cell clonal expansions. Through RNA profiling and VDJ together, we'll touch upon impacts on clinical diagnostics and drug development. Leveraging MGI's unique DNBSEQ platform, we'll discuss advancements in bulk RNA, TCR and BCR analysis, single-cell profiling, and emergent spatial biology technology for multi-omic detection and cellular immune reactome analysis. Without further ado, let's extend a warm welcome to our first speaker, Dr. Shveta Goyal. She's a staff scientist from Takara Bio, USA. Her interest is targeted NGS with emphasis on immune profiling, especially the B cell receptors repertoire. Dr. Goyal did her PhD in biochemistry from the NUS, National University of Singapore, where she studied protein structures and protein-protein interactions. She has also worked on drug discovery and precision medicine projects to find the treatment trajectories for cancer patients. Her presentation today is titled Smart and Sensitive Full-Length BCR Immune Profiling. Let's welcome her.
Thank you, John, for the intro. Please let me know if you can see my screen.
Yes.
Okay. Perfectly clear. Okay, thank you so much. Thank you for today's webinar. I'm Shveta Goyal, and I'm a staff scientist at Takara Bio USA. Some of you may know Takara as by the name of CloneTech. And the title of my talk is Smart and Sensitive Full-Length BCR Immune Profiling. Before I start, let me give a brief introduction about Takara Bio. So, oops. Can you still see my screen?
Yes.
Okay. So for over 50 years, Takara Bio has been helping scientists by providing innovative solutions and technologies. With its headquarters in Kusatsu, Japan, Takara Bio has worldwide affiliates across North America, Europe, and Asia. And we also have additional facilities. For manufacturing and sales across the globe. Takara Biotechnologies cover extensive portfolio of reagents, instruments, and services, including frontline diagnostics and clinical applications. We provide world-class solutions for cloning, gene delivery, gene expression, genome editing, nucleic acid purification, PCR, protein expression and purification, and a whole lot of it, along with next-generation sequencing. With next-generation sequencing, we provide transcriptomics, genomics, and targeted sequencing application. And immune profiling comes under the umbrella of targeted sequencing. As my talk today is about immune profiling repertoire, so let me give a brief introduction about immune profiling repertoire. So Takara Bio provides a solution for human as well as mouse, T cell receptor as well as B cell receptor profiling solutions. For T cell receptors, we have single cell TCR alpha beta profiling kit and bulk TCR alpha beta profiling kit, which is also compatible with RESUS. We always try to improve ourselves, and for B cell receptors, we have an older version of the kit, which is Smarter Human BCR Profiling Kit, and a newer version with the SmartSeq Human BCR Kit, which has a more user-friendly features. For mouse BCR and TCR, we have recently launched a SmartSeq Human TCR Kit with a more updated version, and we will soon be launching the mouse BCR Kit. Now, the good features about these kits are that they provide flexible input range. User can prepare libraries from 1 nanogram RNA input to 1 microgram RNA input, and there is huge flexibility in terms of the sample type they can use. They can use isolated B cells or T cells, PBMCs, whole blood RNA, bone marrow, or lymph node samples. Our solution has UMIs, which are unique molecular identifier, which can help in the error correction during the downstream analysis. And it has good sensitivity in terms of finding the low abundant clonotypes. We capture most of the isotypes. Like for TCR, we can capture alpha and beta, and for BCR, we can capture all the isotypes for heavy chain and light chain. We provide sequencing flexibility. User can do full-length sequencing where they want to identify CDR1, CDR2, CDR3 regions, or if they are only interested in CDR3, they can use 150 by 2 base pair sequencing. And we provide multiplexing capabilities. User can do 384 samples in a single run. Now, the next question is, why is it important? Why is it important to study BCR immune profiling? So now, with the advancement in the NGS, the high throughput sequencing has opened avenues for BCR immune profiling and to study mechanisms for the development of B cell repertoire. And this in turn helps in the understanding the mechanism for vaccine development strategies. BCR, which is B cell repertoire immune profiling, can also be studied to understand antibody repertoire related to the disease or due to the age, because we understand that with age, antibody diversity decreases or there are more mutations found in the antibodies. Some can track the vaccination course, like changes in the immune profiling after vaccination or diseases. And also, it's important to study minimal residual disease, like what is the number of mutated clonotypes remaining after treatment or before treatment. It can also be studied for organ transplantation and antibody discovery and antibody engineering. Now, B cells are very much diverse, and their diversity is because of different mechanisms. So B cells are made up of VDJ regions, which are in the DNA, they are expressed at discontinuous genes. Now, and only one of the V and D and J region can combine in the mature lymphocyte. So this recombination event can lead to almost 6,300 possible recombination events for heavy chain and 350 possible recombination events for light chain. Apart from that, when the recombination happens, there are nucleotides which can be inserted or deleted at the junction region, which results in junctional diversity. This further increased the diversity of heavy chain and light chain to a huge amount, like 6 million for heavy chain and 0.3 million for light chain. Apart from that, when heavy chain and light chain come together, the total possibilities for the B cell receptor can increase to 10 to the power 12. And apart from that, there are also somatic hypermutations which can result in increasing the BCR diversity. Now, immune profiling solution from Takara Bio provides the solution for this challenging diversity and has been used by scientific community across the globe. And it has established reputation of a high-ranking method as described in some of the scientific reports published in recent years. One of the study published in Nature compared 9 different TCR profiling methods, and the result from that study validated Takara Bio's method outstanding performance in terms of replicability, reproducibility, sensitivity, and specificity. One of the important features of the solution is providing end-to-end solution from library preparation to data analysis. I don't know what's happening. It jumps to the next slide. Now, library preparation takes only approximately 5.5 hours with a very little hands-on time of approximately 2.5 hours only. After generation of the FASTQ files, the data can be analyzed using Cogent Immune Profiler, and user can also get the essential graphs made using the Immune Viewer application. Now, the sequencing time depends on what platform or what read length a user are using for the sequencing. Now, the Cogent Immune Profiler is a software which we provide free of cost, and user can use it for the data analysis. This can be used for Windows as well as Mac, and user doesn't need to have a whole lot of bioinformatics skills to do the data analysis using Cogent Immune Profiler. The great general workflow is that Cogent IP, Cogent Immune Profiler, aligns the FASTQ files generated after sequencing to reference a sequence, which is basically TCR and BCR reference sequences. And at this point, short reads or ambiguous reads are filtered out. Followed by this, the files are analyzed for UMI-based analysis using MyGIC and the clonotype calling using MyXCR. And the CSV files are generated, which actually are basically summary reports, which have the information of QC statistics, clonotype report, and these CSV files can be directly used on Immune Viewer for plotting the graphs. With Immune Viewer, users can get the CDR3 length distribution for the nucleotide bases as well as for the amino acids. These give us important information because in some states, in disease states or like in age groups, these may not be bell-shaped. It could be skewed to left or right. We can also get VJ distribution plots and chord diagrams, which can tell us the information about the distribution of V and J genes in the samples. Now, this is like a very straightforward and simple workflow where RT is DT primed, and during the cDNA synthesis step, the UMIs are incorporated along with the PCR handles. Because of this step, we don't need lots of primers to amplify V regions. We only need one universal forward primer and the PCR primer, reverse primers, which are bound to the constant region. And after the nested PCR, we get the final library. Now, this final library can be converted to the MGI specific library by MGI Easy Universal Library Conversion Kit and can be sequenced on the MGI platform. This is how the library structure looks like. We have the UMI followed by the leader sequence and VDJ region, and the CDR3 region. As I said before, user can, if they are only interested in the CDR3 region, they can do 2 by 150 base pair sequencing. But if they need the full length information, it's the new MGI platform is really a good option. And I believe that CDR1 and CDR2 regions also give great information about antibody, antigen affinity. Like in the more recent studies, it was found that from the acidic tumor environment, out of 4 out of 5 mutations were found in CDR1 and CDR2 regions. And with the more high throughput sequencers like MGI, I think users can benefit the full length information. This is some of the data from the human BCR kit. As I said before, we provide the flexibility in the input range. So these are the libraries prepared with 10 nanogram PBMC RNA and 1,000 nanogram, 100 nanogram and 1,000 nanogram PBMC RNA. And we can identify all the isotypes in different RNA inputs. And there is flexibility in the sample type also. Here's the data from PBMC, whole blood, and B-cell RNA. And as we understand, the diversity of the sample changes the clonotype counts in each. The sensitivity of the kit is really great. We see the linearity of detection in the spike in data, where we have spiked in cell line RNA into PBMC RNA up to 0.1 picomolar picogram concentration, and we can still detect the spiked in RNA from the cell line into the PBMC sample. The flexibility of the sequencing, like from 600 cycle or 300 cycle, there is more than 90% clonotype overlap for CDR3 regions. And also, as the workflow is very simple and very straightforward, the libraries are automation friendly, and one can miniaturize those too. So I'm here showing the data from full volume as well as half volume libraries preparing using Mantis liquid handler, and the performance across full volume and half volume is very much comparable. We have also compared our human BCR kit with the competitors, and we find that our performance is, as compared to the competitor, is really great. We found more clonotype count as compared to the other kits available in the market. Now, this is the data which we have generated using MGI's sequencer, and we found that the quality of the data from MGI sequencer is really good, with very less error rate, and Q30 score is very, very good. And the overall error rate is less than 1%, because as the sequencing length increases, the quality drops, but here we are not seeing that drop. This is some of the data which we have generated using MGI platform, and this is the collaboration data which we have done with FDA. It's still ongoing. Here we have prepared the libraries from 2 monoclonal cell lines, Dowdy and CA46, and made a mix of that at 3:1 ratio, and then spiked in that into PBMC RNA at 10% and 1% ratio. And these libraries were prepared using the Takara's human BCR kit. And when we sequence those on MGI's platform, we see that read mapping is very good. More than 95% of the reads are mapping to the reference sequence. And also the molecular identifying groups are, it shows that as the expression of the sample is high, we find more molecular identifying groups. The clonotype counts identified for PBMC heavy chain and light chain also shows that when we spike in monoclonal cell lines at different concentrations, the clonotype count of the sample decreases, but with the pure PBMC RNA, the clonotype counts identified are more. So this is how the performance of the data is when the cell line RNA is spiked in the PBMC RNA. So DAUDI and CA46, they are spiked in at 3:1 ratio, and we see that the same sort of trend in heavy chain samples. DAUDI and CA46 is actually in the same proportion as in the cell line mix, but in light chain, it is actually not the same. CA46 is more and then DAUDI. And as I was showing in the previous slide, expression of CA46 is higher, and that's why we are seeing that kind of trend. We see the same sort of analysis using the core diagrams, where the monoclonal cell lines in the core diagram show only one sort of V and J gene, while when we mix it, we see the proportion of DAUDI and CA46 in the same ratio as they are mixed. And in the light chain also, we see DAUDI and CA46 as single arc, but when they are mixed, it is in the 3:1 ratio. Now, the data generated from Cogent IP can also be used for further downstream analysis using some other softwares like ImmuneArc. So these graphs were generated using ImmuneArc, and it shows the relative abundance of clonotypes. So in the monoclonal cell lines, because there is only one clone, we'll see only one hyper expanded clonotype. But in the PBMC RNA, where we have spiked in 10% and 1% of the cell line, we see more of hyperexpanded clonotype and less of the small. But when there is no spike in, hyperexpanded clonotypes are less, but rare small and medium clonotypes are more. So it has more diversity. And we can also use the data to see the repertoire overlap. And because these are the PBMC RNA, the same background, and we see more clonotype overlap between those samples. With this, I would like to conclude that Takara Bio's BCR immune profiling solution is compatible with MGI's DNBSEQ sequencing platform. It checks all the criteria for sequencing quality, read mapping, and clonotype identification for all the isotypes. We can capture entire BCR variable region for heavy chain as well as light chain isotypes, and it provides high sensitivity, reproducibility, efficiency, and we can detect low abundant clonotypes in the different input ranges. We provide unique dual indexes and so multiplexing ability to 384 samples, and there's a flexibility for sequencing on different instruments. And this features automation-friendly, easy, and user-friendly workflow. With this, I would say that's good science. And if you have any questions, I will be happy to answer those.
Thank you so much, Dr. Goyal, for your insightful presentation. At this juncture, we invite any questions from our audience to our panel speaker. Please feel free to use the Q&A in your right-hand side. There's this panel. You can type in for questions, or you can chat also if you have any other questions outside of scientific realm. Thank you. So we learned today nicely from the sensitivity of Takara immune profiling assay running on the DNBSEQ panel platform, and demonstrated this vast complexity of B-cell receptor repertoire. And actually, it's so nice to see that the necessity to capture all these clonotypes accurately, right, with the sensitivity method. So perhaps I'll just throw in one question here. You have a slide that's comparing your BCR clonality, right, with a competitor. So may I ask, what is the why do you think that Takara's is more sensitive to get more clonotype counts here?
I think the main reason is we have kept our heavy chain and light chain separate because of the different lengths of heavy chain and light chain. So if you see that most of, if we look at the read distribution, which I'm not showing here, most of the reads are just going to light chain. So that is one thing. Another thing is our chemistry. the way our UMI incorporation and our enzymes work, we identify that makes our chemistry more sensitive.
Great, thank you. Well, maybe I'll just ask another question since we still have some time. Could you explain a little bit of adding the UMI, because I see that's the evolution of your kit's versions, right? You have UMI added in, comparing to also UDI. Maybe give us some more insights on how this is developed and what it brings to value.
Yeah, so UMIs are actually added at the RT step. So we have the smart chemistry where when we are doing the RT, the reverse transcription, and preparing the cDNA, at that step, we do the template switching, and the template switching adds the extra nucleotides. And these extra nucleotides then helps in the addition of UMIs by the reverse SmartScribe reverse transcriptase enzyme. This UMI is actually called a unique molecular identifier, which each transcript will get one UMI. Now, if there are some sequencing errors or PCR errors, these UMI can help in correcting those during the data analysis part. Now, moving to your next question was addition of UDIs, right?
Yep.
So UDIs are basically in the past, when we were using HD indexes, those indexes, like they were single index, right? And there were more chances of index hopping. By UDIs, we minimize that index hopping problem. And also, you don't need multiple tubes, and it helps in automation also. So it's like one single tube which adds the UDI, not the two tubes which you need for HD indexes.
Okay, great. Thank you so much. Let's move on to our next speaker. Okay, our next speaker is Dr. John Beckford. He's also a scientist from Takara Bio, USA. John is a scientist in Takara. He earned his PhD from the University of Southern California, where his research focused on the immunomodulatory effect of epigenetic therapy in the acute myeloid leukemia settings. So let's dive into his talk, which is titled Unbiased, Sensitive, and Accurate TCR Analysis with the SmartSeq Human TCR Kit. Let's welcome him. John, I think you're muted.
Can you hear me now?
Yes.
All right, definitely. So thank you for the introduction. My name is John Beckford, and tonight I'll be talking about unbiased, sensitive, and accurate TCR analysis with the SmartSeq Human TCR Kit with UMIs. So first, I'm going to give a little overview of the SmartSeq Human TCR Kit. So the chemistry that powers this kit is the same underlying chemistry shared by the SmartSeq Human BCR Kit, actually, where we generate cDNAs with a smart sequence, a universal smart sequence, and a UMI at the 5' end of our cDNA. And then we leverage that universal 5' sequence and then the universal 3' sequence, which is actually in the constant region, to further amplify our TCR transcripts from the cDNA, and an additional round of PCR to further enrich our TCR transcripts and add UDIs on top. And then in doing so, we generate a sequencing-ready library that can then be converted to the MGI format and then sequence on the DNBSEQ platforms, right? So some of the key features of our kit is, first, unbiased TCR amplification, unique molecular indexes, unique dual indexes, flexible sequencing, so full length or CDR3 only sequencing, and compatibility with our immune profiler software. So together, all these features allow for the capture of the full spectrum, the full diversity of the T-cell repertoire, both chains, the alpha and beta chains. And one important feature that I want to quickly touch on is compatibility with rhesus macaque samples. So yes, this kit is called SmartSeq Human TCR with UMIs, but it also works with our rhesus monkey samples as well, right? So next, I'm going to get into some data that was generated directly on the DNBSEQ G400 platform. So first, I'm just going to show the consistent clonotype capture on the DNBSEQ. So right here, we have two independent libraries that were generated with the same input amounts and sequence on the DNBSEQ, downsampled to about 10 million reads. And as we can see here in our slides, in our graphs, we get roughly the same number of TCR alpha and TCR beta clonotypes, where the blue bars represent the TCR alpha clonotypes and the purple bars represent the TCR beta clonotypes, right? And then next, I then want to get into confident clonotype identification on the DNBSEQ G400 platform. So one of the real important things that we need to do with TCR sequencing is correct clonotype calling, right? Because of the random nature of the CDR3 region, any sequencing error can be passed off as a real clonotype when in fact it's actually a sequencing error artifact, right? And these sequencing errors can be accumulated as one sequences deeper and deeper and deeper. But with our chemistry, with the addition of UMIs, we're able to mitigate the effects of over-sequencing our libraries and filter out those clonotypes that may have been generated by PCR or sequencing errors. So if you look at the two graphs that I have shown on the screen here, we have one curve which is which was generated with UMI thresholding, and then another curve which was generated without UMI thresholding. And these are curves that plotting the clonotype count across a sequencing depth range from 100,000 reads all the way up to 10 million reads. So as we can see, for both the TCR alpha and TCR beta graphs, you see that without any UMI thresholding, you will see additional clonotypes on the order of a couple thousand more clonotypes. These may be real clonotypes, but it's a very good chance that these clonotypes were generated by PCR sequencing errors. And because of the way our chemistry works, we're able to filter out those low confidence clonotypes that may have been generated by error, right? Leaving the end user with data they can trust for their publications and their studies, right? And then next, I want to briefly touch on the capture of the full spectrum of diversity of the TCR alpha repertoire on the DNBSEQ G400. So on the left-hand side, I have a core diagram of all the different VJ pairs that appeared in our data. So it's a lot to look at, but the main message you get from this diagram is that you do capture like the full spectrum of TCR alpha repertoire diversity on the DNBSEQ G400 platform. And on the right-hand side, we have all the V and J genes that appear in our data set. So if you can quickly scan across both the V distribution graphs and the J distribution graphs, you actually see every single V and J gene that's currently in the public databases represented in our data set right here. Again, just showing that with our chemistry, you can capture the full spectrum of the TCR alpha repertoire diversity, right? And then the same situation with on the beta chain as well. So on the left-hand side, again, I have the core diagram showing all the different V and J gene pairs. And then on the right-hand side, I have the graph showing that the V gene distribution and the J gene distribution. And like before, you can see every single V and J gene represented in our data set, indicating that our chemistry is really good for capturing the full spectrum of TCR beta diversity, right? And then the last piece of data that I want to get into today is the use of, again, the DNBSEQ G400 platform to capture the full spectrum of TCR diversity as expressed by CDR3 length. So I don't know if we're like familiar, but within the TCR repertoire, the clonotypes that are present have a range of CDR3 lengths in terms of amino acids, right? So with this chemistry, we can see all the short CDR3 regions and all the long CDR3 regions. Right now, it's a normal distribution, but in your own studies, when you might have a disease, when you're doing profiling on a sample from a disease state, you might see a shift towards the left or towards the right to indicate something going on. But regardless, you're able to capture the full spectrum of CDR3 lengths on the DNBSEQ G400 platform, right? And then to conclude, the SmartSeq Human TCR UMI kit is compatible with the DNBSEQ G400 platform. This is evidenced by reproducible TCR capture, unbiased analysis of the TCR alpha and TCR beta repertoires, and cogent NGS immune profiler and immune viewer compatibility. And that's good science, and that's all I have for you today. Any questions?
Thank you so much, Dr. Bedford, for presenting us how the SmartSeq Human TCR kit is a complete solution for unbiased and sensitive, accurate TCR analysis. We have one question I see in the audience. I'll just read it out. So, are there options for examining TCR gamma, delta, and KIR receptors and using long-read sequencers?
At this time, we do not have solutions for long-read sequencing and gamma, delta, or KIR. We're just TCR alpha beta on the MGI platform.
All right. Thank you so much, Dr. Beckford.
Absolutely.
Let's move on to the next speaker. Thank you. Right. Our next speaker is Dr. Huo Xiaojing, Senior Product Manager from Singleron APAC in Singapore. Dr. Huo has received her PhD in cell and molecular biology and was trained in computational and system biology. She has extensive experience in immune profiling, clinical development, deep immunomics, platform development, bioinformatics training, and product management. Today, she's going to present her work titled New TCR Profiling Solutions from High Sensitivity Single Cell Profiling to High Throughput Bulk Sequencing from Singleron single cell assays running on DNBSEQ platform. Let's welcome her.
Okay. Thanks, John, for the nice invitation and introduction. So I'm happy to share our new solutions for TCR profiling from the high sensitivity single cell profiling to high throughput bulk screening. So before I step on the immune profiling topic, I would take this opportunity to give a quick overview of our company, Singleron Biotech, and also a brief introduction of our single cell analysis techniques, where we can address and we can see how we utilize those new solutions to advance the immune profiling at the single cell level as well as the bulk level. So Singleron is an innovative molecular diagnostic company dedicated to applying groundbreaking single cell analysis techniques in research, clinical diagnosis, drug development, and health management. So the company was incorporated in January 2018, and currently we have offices, laboratories, and manufacturing facilities in Cologne, Germany, Nanjing and Suzhou in China, New Haven in USA, and Singapore. So for our innovative single cell sequencing solutions, we provide the full suite of products, covers the end-to-end workflow for the single cell analysis. So at the beginning, we need the very good sample preparation to achieve the good quality of single cell suspension. And as a solution, as the products, we have the automated tissue dissociation equipment, which is Python and Python Junior. Incorporate with it, we also have the tissue preservation and the tissue dissociation kits. Then with those nice single cell suspension, we wanted to get the single cell partitioned in our microwell-based chip and work through for the next step, cell processing and the library preparation. So as automation solutions, we have Matrix for the single cell processing, which allows us to loading the cell suspension to the chip. And for the library preparation, as well as the multi-omic solutions, we have a variety of kits available for different usage, including the VDG kit, the S-Circle kit for full length, and the Akira code TCR for bulk. With the good quality of the library constructed, we proceed for the sequencing, and currently our libraries are compatible to all the short-read and long-read sequencing platforms, including the MGI solutions. After the data achieved, we also have a full set of bioinformatics analysis support by providing the Telescope bioinformatics analysis software with the AI-driven MapSuite software set, and also the Ecosys and MapDB database, which has curated hundreds of millions of cell data over there with integrated clinical features. So with this well-streamlined single-cell sequencing workflow from the tissue processing all the way down to analysis solutions available, I would like to highlight our core technology, the scope chip here a bit. So basically this scope chip captures the single cells by partitioning single cells into hundreds of thousands of microwells on the chip. And afterwards, an axis of barcoding beads is added to the microwells of the chip. Then after cell lysis, the barcode beads, each with a unique cell label, which is the barcode, will capture mRNA by binding to the poly tail on the mRNA. And then those beads are subsequently collected from the microwell chip, followed by reverse transcription and PCR amplification. So the end result is the next generation sequencing library, which is compatible with Illumina sequencing instruments, and also can be run on the long-read sequence and MGI sequencing platform with minor modification. And about this chip, by selecting different chips for the different throughput and the microwell sizes, scope chip can analyze 500 up to 30,000 single cells simultaneously and accommodate different cell morphologies and sizes. So based on the scope chip technology, we have developed a unique collection of single-cell multi-omics solutions. Basically, we have the JAK-scope for the mRNA profiling for gene expression at the single-cell level. And we have developed FocusScope, which performs single-cell analysis of both gene expression profile and the point mutations, fusion genes, or intracellular viral sequence simultaneously. Also, we have CySeq solutions for gene expression and cell surface protein and epitope profiling. A unique one, we have the promoscope for gene expression, and the glycosylation abundance detection. The Dynoscope, which can study the RNA dynamics by differentiating the new transcripts from old and enable the measurement of the kinetics of RNA transcription and degradation. And this solution can be applied for both in vivo and in vitro studies. And of course, we also have the ClinDex for the sample multiplexing. And the last I would like to mention for S-Circle, JAXCOPE VDG, as well as the Accurate TCR solution, we have the immunoprofiling solutions. Now I hope you get a good understanding of the overview of single RNA as well as our core technology, and let's dive deeper into the immune profiling and the TCR sequencing solutions. So basically, we provide a comprehensive immune repertoire profiling solutions. At the single cell level, we are able to capture the individual chronotypes associated with the phenotypic profiling, the mRNA profile plus VDG, and specific for the selection of the VDG, we have JAXCOPE VDG kit for focus on the CDR3, and S-Circle for the full-length immunoreceptor. At bulk level, we're able to profile TCR repertoires at population level with multi-samples at the same time, with the support of the Accurate bulk TCR library kit. So a little bit about why we are interested in immunoreceptor repertoires. As the key components of adaptive immune system, T cell and B cell takes the antigen-specific defense for our body. The specific antigen recognition is achieved through the receptors that express on the cell surface. And each cell only expresses one type. This provides a nice material for us to identify the cell together with its function. However, the complex of those antigen receptors comes from the huge variety of surface receptors, and this variety was generated from the gene rearrangement and somatic hypermutations. And to profile the high diverse repertoire, we may understand some facts about the receptors. So the two major factors, gene rearrangement and hypermutations, are mainly on the VDG regions for the receptors, and CDR3 is the most viable one. And we also observe the features of CDRs within the viable domain. CDRs bind to the epitopes of antigen. And most of the sequence variation are found in the CDRs. Specificity and the diversity largely rely on CDR3. So as the based on those facts, we say that in the TCR BCR repertoire analysis, the sequence of CDR3 has the most important chronotype information, and most of the immune profiling experiments focus on the CDR3 region. So based on those above interests, we have developed two single-cell immunoreceptor profiling kits to fit the different application needs. So more for research purpose, we have the JAXCOPE single-cell VDG library kit, which could support both human and mouse VDG profiling, and it collects the RNA library and VDG library, which is CDR3 focused for the profiling of single cell. The technology is to utilize the special designed probes on the beads to capture all the TCR or BCR consonant regions. And the applications could could provide support to the tumor research, immune diseases research, interesting for a lot of biomarker screening, antibody evaluation, as well as the infectious disease research, which can help us better understand the mechanism of drug actions and also monitor the immune dynamics during the infection. In parallel, we have the full-length immunoreceptors, where we can address more therapeutic development needs. So for now, we have the humans, we support the human cells profiling, and the libraries will generate for RNA and full-length immunoreceptors. Technology-wise, the mRNA capture is still through the poly-A tail, and to achieve a high-quality full-length immunoreceptor, we use the circularization of cDNA to bring the VDJ region closer to the sequencing primer. And as a application similar to VDG kit, some of the disease monitoring, the immune microenvironment research is also applicable for this kit. Also more for clinical usage, the autoimmune diseases, transplantation, and therapeutic development will get very good support and benefit from this solution. So if we look at the principle of the VDG library kit, we have the beads with two probes, two kinds of probes, the poly-DT to capture mRNA, and the alpha chain, beta chain probe to capture the constant region on TCR. And after all, we have two libraries, the mRNA library for gene expression and the CDR3 targeted TCR BCR library. So the next question is, how is the performance for this new solution for the CDR3 focused and the TCR probing at single cell level? So we do a comparison study between the supplier X solution and the single run JAXScope VDG kit. So as a result, we see a good capture rate of the paired alpha chain and beta chain with over 80% of the cells, which can identify with both alpha chain and beta chain. And if we look at the reads mapped to VDG, alpha chain and beta chain, VDG solution give a higher percentage of the reads. So from here, we see the designated TCR probes provide an increased specificity to capture the TCR, and as a result, it shows the higher capture efficiency for TCR. Then with this result downstream, we can do tracing of B-cell, T-cell lineages, measure the diversity, and align clonotypes for the downstream experiments. And if we split the VDG, we split the single run libraries into two and do a comparison between the sequencing platforms, we do see some comparable results. So this slide shows the RNA library results for at the panel A, we use the feature selection or the identification of informative genes, which play a key role in the single cell studies as the indication of the performance. So in this evaluation, it shows the distribution of features, the features which are the detected genes, and the number of counts for each cell. And in the different color dots, we see the sequencer I and MGI give the very similar result, shows the similar distribution. And next, the further UMAP analysis shows the sequencer I and the MGI have the cells clustered altogether very well. It demonstrates the similar gene expression pattern between the two platforms. And last, if we evaluate the ability of cell type detection, we have annotated those cells into their cell types using the single R and with a public multi-model reference atlas of the circulating human immune system. Then we also see at the cell composition, their both platforms shows very similar results. And we also look into the VDG library, look into their performance on the different sequencing platforms. So in this bar chart, we use the black framed box to show the double chain detected cells, which are the light chain and heavy chain paired cells for B cell, and the alpha chain, beta chain paired for T cell. On stack on it, it's the this gray framed bar shows the single chain detected cells, and in the panel A and B, we see that the number of clonotype cells and the number of clonotypes detected shows very similar result between the two platforms. And in the B cell, we show slightly higher detection rate from the MGI sequencing. So after the result about the single run JAK-scope VDG performance, we also take a look at the comparison between the full length solution, which is between the single R S circle and the supplier X. And with the percentage of cell with alpha and beta chain paired detection, we see nearly 80% of the cells get both alpha chain and beta chain paired. So and with the mapping rates rate, S circle shows slightly higher percentage of the usable rates. So this result has indicated those comparable capture efficiency for the TCR, and also a high percentage of the cells with full-length TCR detected and assembled. So here I would like to conclude the single-cell solutions with a summary, highlight our offerings. So basically from the result, we see a good performance of the two single-cell immunoreceptor sequencing solutions. They both provide high discovery rate for the immune receptors, and both of them get over 80% of the cells having both alpha and beta chain pairing. And overall, with those two kits available, we offer the comprehensive and flexible offerings with the CDR3 focused clone typing or full-length TCR profiling using S-circle. Species-wise, mouse and human both are kits are both available, and manually loading loading on the chip or automated using our matrix instrument are both available. And also for the downstream sequencing, we have proved the consistent results between different sequencing platforms. And the last, both kits are very easy to use. One kit includes all the reagents for the RNA and the immune receptor library preparation. And overall, it's a well streamlined workflow from the very beginning of sample processing all the way to the bioinformatics analysis tools available. So with this deep dive into single cell level TCR profiling, we're able to identify those TCR candidates with paired chains detected. And we're able to evaluate the immune responses by monitoring. And also with both RNA library and the VDG library available, we're able to, and simultaneously getting from the same cell, we're able to match TCR profile with the phenotype at the single cell level. So the next question is that with this largely facilitate the function and therapeutic potential candidate discovery, then how do we further validate those discoveries at a large cohort or at the population level? So we would like to also mention the high throughput bulk TCR profiling solution, where we're able to do the high throughput validation, screening of hundreds of samples from a patient population, and also observe the disease-associated TCR sequence by comparing and identifying the common motifs in multiple samples. So our AccuraCode TCR library construction has provided this solution with a time and cost saving manner. So basically the technology is we transfer the single cell barcoding technology to improve the throughput of bulk sequencing library preparation. And in the workflow, at the beginning, we place the cells or the samples at each well in the 96 well plate. And and on top of that, we can place the well indexed primers into each well. After the cell lysis, the mRNA captured in each well will be indexed with the well-specific barcode. Then we are able to multiplex or merge all the samples, all the mRNAs collect from those 96 wells all together into one tube and do the downstream RT-PCR, TCR amplification, as well as library preparation. And at last, we also provide the easy-to-use bioinformatics pipeline to analyze the data, including demultiplex the sample into the 96, then map the TCRs, and the last to calculate the clonotype. So the wet lab part workflow for 96 well TCR sequencing library preparation will only take 7 to 8 hours altogether, and the library preparation process will be done in one tube for all of them. And of course, if there's liquid handler available, this workflow is totally okay to be automated. And at this new solution with the multiplexing of library preparation, we would like to see how is the performance. So we tested the performance on the PBMC into many wells and utilized the UMIs counts mapped to alpha chain and beta chain to see the consistency across the replicates. And in the alpha chain, we see around 20% to 30% of the capture rate, which is comparable to the benchmark results. And for the beta chain, we observed the 40 to 45%, which is consistent across all the replicates and also consistent to the benchmark. So next, we're also looking at the sensitivity of the clonotype detection on this kit. So we use the CCRF, which have the only one known clonotype, and to spike into the human PBMC T cell pool. And as a result, we see the detection limit is from 1 cell out of 10,000 cells, we are able to detect them. And the linear region of detection is on the 0 to 50% of spiking rate. So in this region, we see a spike in CCRF number and the chronotype frequency are linear correlated. So at last, I would like to conclude my presentation with highlight the our offerings for those new TCR profiling solutions. So at the single cell level, we provide this multi-omic solution, and it's flexible for chronotyping, utilize the CDR3 focused VDJ kit or full-length TCR BCR profiling. Both kits has shown the high recovery rate for immune receptors and the high pairing rate of alpha and beta chain. And with those kits, both RNA and TCR profiling, we're able to achieve in the same cell, and as flexibility of the choice of the sequencing platforms, we demonstrate the comparable result from different platforms. Then for the bulk TCR solution, we provide this high throughput TCR-seq solution, which is the AccuraCode TCR kit. It provides the high sensitivity and consistency at the many samples in multiplex. And also through this multiplexing, we provide this solution as a time and cost saving way for the library preparation. And the last, I would like to thank you very much for your attention. And you are very welcome to follow us in the LinkedIn and subscribe our monthly newsletter. Thank you.
Thank you, Dr. Huo, for the enlightening talk. It's great to see the single cell technology, and also now we understand that you have a full length in single cell plus your bulk immune profiling. Like, it's quite comprehensive. Thank you so much. There is now a question coming from our audience wondering which software were used for the analysis. I've seen you presented quite a few software packages or pipelines. Would you care to share?
So basically, we are using our in-house developed software, which called Celliscope. Let me try to jump it into the. So it's Celliscope Bioinformatics Analysis Software. So this is a comprehensive toolkit, which include the almost all the pipelines for our single cell solutions, including the module for the VDJ sequence analysis for S-Circle, as well as for the AccuraCode TCR. So it's free offering on GitHub.
Thank you, Dr. Huo. So yeah, as I mentioned, I think you've shown some of the results is the JEC-scope that we've ran with your provided libraries on the MGI sequencer. I'm sure we are happy if you are happy to collaborate again. We're happy to run the S-Circle kit as well, so we can see how the single cell level, because we have the parent 300 right on the G400 platform as well as G99. I think it would be a good match.
Oh yeah, that's good. I think of course we're happy to, and it's good we can continue this chat offline. Yeah.
Yep. Thank you so much. Thank you.
Thank you.
So to our next speaker. Yes, so yeah, it's my pleasure to introduce our fourth speaker, Dr. Sean Huang. Dr. Huang is the Director of Lab and Services at Marexis, which is a which provides a portfolio of NGS services with high quality and cost effectiveness in Singapore, actually in the A-Star Biopolis campus, right? Dr. Huang held various R&D and leadership positions, including in Thermo Fisher Scientific and Veritas Laboratory. He obtained his PhD from NUS and was an early career researcher in IMCB A-Star. The title of his talk today is Unlocking the Power of Single-Cell Analysis. Let's welcome him.
Hi. Here's the screen.
Yes.
Okay.
Perfect.
Yeah, thanks for the kind introduction. Good afternoon, everyone. My name is Sean. Today, I'm honored to be invited here to share with you some of our recent experiences with single-cell sequencing on the DNBSEQ sequencer platforms in Repertoire, and how Repertoire's genomic sequencing services can turbocharge your single-cell research and unlock the power of single-cell analysis. So for who those are not very familiar with Repertoire, here is the overview of Repertoire at a glance. About 20 years ago, in 2002, our co-founder, Professor Tu Heng Feng, and his team in the lab of NUS demonstrated the proof of concept of detection technology. And 12 years later, Repertoire expanded out from A-Star. Today, Repertoire is the global leader in the microRNA-focused molecular diagnosis. So we have a proprietary and patented qPCR platform technology that can detect and quantify the microRNA in the blood. And we also have a strong pipeline in oncology, infectious disease, and cardiovascular diseases. We have built end-to-end capabilities in R&D, manufacturing, and also clinical diagnostic services. We are a small company and is venture-backed, so we have a strong revenue and cash positions. With more than 180 million USD raised. Throughout the years, we invested very heavily in our internal R&Ds as well as build our capabilities. We have been growing from Singapore to the world, and as we are headquartered in Singapore, here at home we have a very strong R&D capabilities with 3 research labs and 1 clinical labs right here in Biopolis, Singapore. We also have Industry 4.0 manufacturing plants at the west side of the island, as well as global regulatory affairs that overseas markets. Outside Singapore, we have R&D and GMP manufacturing facilities, as well as clinical lab in China, and in USA, Japan, and Philippines. We have sales office and also R&D activities, as well as clinical lab. Back to Singapore. And the Repertoire's precision genomics BU is relatively new and established a couple of years ago. And this BU really expand our capability from a microRNA company to high throughput NGS. Eventually, we are aiming to establish a multi-omics portfolio to support our internal R&D as well as researchers in the community in Singapore as well as overseas. And as you can see, this is a picture of the new genomic lab and the men and women that make it happen. And more than 50% of us have advanced degrees, master and PhD. And we also have a range of sequencers from a low throughput to higher throughput, from 20 GB per run all the way until the highest throughput in the world, which is 80 terabytes per run, MGI T10, which you also can see at the back of the picture here. And most of our sequencer are using the DNBSEQ technology. With the T10 at Repertoire is really a powerful piece that can turbocharge any large-scale research that requires a lot of sequencing, such as a single-cell sequencing. So for those who are non-expert in single-cell like myself, here is the examples of the applications of single-cell RNA-seq. And this picture shows one of the applications in the tumor heterogeneity. So as you may know that when tumor progress, from tumor stem from cancer stem cell, it differentiates into different types of tumor cells, which each of them have different properties. Some of them may have a better chance of invade to other organs. Some of them may have resistance to certain treatment, and some of them is better at avoid immune cell detections. That's why after treatments, a lot of patients experience relapse because some of the cells are not being dealt with. So you will have a microscopic view of a lump tumor will look like this. And traditionally, if you use bulk DNA sequencing technology or RNA sequencing technology, all different cell signatures will be averaged out or masked by one another. Only using the single cell RNA-seq technology, we are able to really identify each of the different tumor cells and also the different type of immune cells that will infiltrate the tumor, which a lot of studies actually shown that the type of immune cells in the tumor actually have a huge impact on the tumor progression as well as the treatment response. So back to the sequencing, the single cell sequencing. This is a dilemma, right? Whether we have a project, we have a part of our budget, whether we do a deep sequencing of a few cells or shallow sequencing of many cells. I believe many things that researchers have faced this before. Actually, one group from Stanford University actually published a paper on Nature Communication on this topic, which shows how important this topic is in the single cell research world. And when we sequence a few cells at a deep, at a great depth, you can accurately estimate each individual cell, but you will lack the coverage of entire distribution like this. Whereas if you only sequence shadowing of many cells, you may cover the entire population, but you also at the same time introduce a lot of noise. So this situation, I think, is greatly constrained and limit the power of single cell analysis. And can we do much deeper sequencing with more cells? That depends whether you can find a cost-effective way to generate high quality single cell sequencing. Way to generate high quality single cell sequencing data. And in Meraxis, we have been developing many sequencing strategies for major single cell players, such as 10x bioscience, Pass bioscience. And other than single cell RNA sequencing, we are also ventured into single cell DNA sequencing as well, such as single cell whole genome sequencing. But today, my talk will be only focused on this workflow, which is the 10x genomics library preparation, followed by the DNBSEQ. And here is the overall of the workflow. After the standard 10x library preparation, the Illumina linear double-stranded DNA library can be easily converted to a single-stranded circularized DNA library that is compatible to the DNBSEQ sequencing platforms such as G400T7. So after that, the data can be analyzed by the same 10x genomics pipeline cell ranger. And I will be presenting the data from two benchmark studies. One of them is conducted in Meraxis together with our with the solid tissue samples, and the other is conducted by a dependent group using PBMC blood cells. So in this Meraxis study, the 5 libraries has been previously sequenced on Illumina platform with clients' chosen facility, and the clients kindly passed us these same 5 libraries for us to resequence them in Meraxis on the DNBSEQ T7. Then we give back the data to the customer for them to do a benchmark analysis so that we can have a very unbiased view of how the performance look like. And after that, clients kindly share the data with us, and we have the permission to present the data. So first of all, the first kind of sequencing quality comparison, we always look at Q30 percentage. As you can see, blue is from MGI sequencer T7, and orange is Illumina. Both platforms actually give us very high quality of sequencing data. Q30% is more than 90% for all the libraries and for both platforms. Interestingly, when you look at the percentage of reads that map the genome, we can always see a 5 to 10% more reads from the T7 data set that can be mapped to the genome. As you may know that whatever the reads that not mapped to the genome basically is wasted. And then we look further to other QC metrics like estimated number of cells. They're very, very similar and almost the same. And the median genes per cell also follow the same trend of different libraries. And despite of we have different number of reads from different libraries. Even at this library number 5, when we have less reads from the T7 dataset than the Illumina dataset, we still have higher. We still see a higher percentage of reads that map the genome. So this means that it's not about the quantity of reads. It's definitely something about the quality of the reads that come out from the T7, which we don't know exactly why, but this is it is to our pleasant surprise. Then next, let's look at some QC data metrics. First, this figure A, we look at the distribution of the unique genes and feature, which represents unique genes in M10. And also the uncounting is the number of UMI counts, as well as the percentage of the mitochondria reads, which shows whether the cell is alive or not. Higher percentage of mitochondria reads indicates the cell is dying. But basically, the idea is to look at the shape of distribution that are very, very similar. Now we look at this N feature versus N count. I think you saw the similar feature in Xiaoyin's talking previously. They are highly overlapping between MGI and the Illumina sequencer datasets. In the figure C, you can see the correlation between the N features versus the percentage of the mitochondrial reads. Basically, you see a very similar pattern, and you also see that actually the data, the cell rate is good. We see much more genes in variable cells versus the cells that are dying. And more importantly, we see no difference in downstream analysis. One of the analysis, for example, cell annotation. When the clients did a gross cell annotation QC, where they used a feature more than 200 and the percentage of mitochondria less than 30% to do a cell annotation UMAP, you can see from the two separate data sets from Illumina and MGI actually generates a very similar UMAPs. You cannot even distinguish them, right? And when we overlay them together, it's almost nearly perfectly overlap to each other. And secondly, when we look at the marker gene expression, and here is an example of the heat map of the cellular cell marker from the Illumina dataset as well as the MGI dataset. You can see the pattern of the gene expression is almost similar as well. So as I mentioned before, all this analysis is done by our client. And when they share this analysis with us, they also share their conclusion. So this is a conclusion from a client and a code here. There are no significant difference observed in the base quality scores, the number of detected genes, and the number of detected UMIs per cell between the two. Additionally, in the downstream analysis, the cell exhibits perfect overlapping in UMI, in UMAP, and further indicating the similarity of two data sets. And overall, the finding indicates that there are no significant difference between the sequencing result of Illumina MGI and highlighting their striking similarity in the performance. This same point has been briefly mentioned in Xiaoji's talk previously as well, and I was very happy to see that. And secondly, the second study is from a different group, and I don't have permission to mention some of the names, so apologize for the highly sanitized slides data. And the reason that I want to present this case to you is, as a scientist, we are always very happy to see our findings independently validated by another group. This really gives us more confidence, but sometimes we are just so critical to our own work. So yeah, those results are presented in a slightly different way, but they are telling the same message. So in the figure A, it shows the Q30 of the libraries from two platforms. The blue one, sorry for the quality of the slides, the picture. The blue one is from G400, and the red color represents the brand A, and we all can guess which brand it is. And they're all very similar and all above 80% as expected. And when we look at a different part of the library, they are very, very compatible and all very high at a very high quality. When we look at the saturation curve, so as the median gene per cell curve, the two libraries from two platforms are always overlapping, very close to each other. Then I think you see this plot many times. The distribution between N features versus DNA count, and in the gene expression libraries, they're very highly overlap, as well as the capture library, which is the feature barcode library. It also overlaps pretty well. And most importantly, when we look at UMAP of cell annotation, all four libraries shows very similar UMAP from both brand A and MGI G400. So take-home message is very clear and simple. The DNBSEQ sequencing platform is highly compatible with the 10x genomics single-cell libraries, and it provides a cost-effective way to generate high-quality single-cell data. I think you by now probably have been convinced about that. So if you don't want to get your hands dirty on the sequencing and want to focus on science, if you want to get even more bang for your buck, I have a bad news for you. So Merck's Genomics has recently launched a single-cell RNA sequencing service. And here are a few unique starting points. Here we are located here. Our facility is located here in Biopolis, Singapore. So your sample won't be sent to overseas to avoid any risk of logistics. And we provide unbeatable sequencing quality, as I just shown, as you have just seen by. And last but not least, we can provide comprehensive bioinformatic analysis or tertiary analysis, so that we can make more sense out of the data. And if required, we can also provide a sample to manuscript feature and to unveil services if you don't have time to. For that, and if we want to focus on other part of the science. And things are just getting better towards the end of the year. So we are launching an exclusively priced match campaign for our services. So what you need to do is show us your price that you get from other place, and you will get up to 30% discount off the price that you showed to us. And please take a picture and also screenshot, maybe also screenshot of this QR code, and how you can participate and take advantage of this campaign. Then, of course, in Merck's, we are not just a sequencing service provider. We drive breakthroughs from collaborations. We are very experienced in and also welcome in any kind of research collaborations with public research institutes. We also welcome any co-development of product or services with business departments. So finally, if you want to know more about Merck's and or have any new research ideas, feel free to reach out to me at seanhong@repertoire.com. And if you're in Singapore, the coffee's on me. We can always discuss the ideas. Thank you very much.
Thank you, Dr. Huang. Thanks for sharing your expertise. I think it's a very unique perspective that we not only have vendors and also scientists, but also coming from a service lab, right? So we're very pleased to hear that actually DNBSEQ sequencing library, sequencing platform is the preferred choice, or at least it's very much comparable and sometimes even better than other sequencing platform, so coming from both the 5' RNA-seq as well as the VDJ and feature barcode from the 10x genomics example. Thank you. Any questions? All right. Would your result be shared with us? Yes. So I just might as well take this chance. We do have the presentation, and with the permission from the speakers, we'll be able to share with our audience. Please leave your contact detail on the chat box.
Yeah, although you see those are strictly confidential in my slides, I think that's come with our company template. So just you can ignore that.
Right. Derek Wang mentioned that you are TFS colleagues. I hope. Oh, that must be Thermo Fisher then. Right. Okay. So thank you so much, Sean. I think we're going to move to the last person, which is actually coming back to the BGI team, right? So let's introduce our next and last speaker, Dr. Heng Yang. Dr. Heng received his PhD in neuroscience at the University of Groningen in Netherlands in 2021. His main work is on innate immune memory and transcriptional profiling of microglia, working with Professor Bart Eggen. Dr. Heng is currently the R&D scientist at BGI Research in Shenzhen. His main research focus is in spatial multi-omics methods development. His team is responsible for the development of protein and RNA co-detection methods based on the StereoSeq spatial technology. The title of his talk today is High Spatial Resolution Co-mapping of the Whole Transcriptome and High-plex Protein using StereoSeq. Let's welcome him.
Okay, thanks, John, for the introduction. So today do you have my screen now?
Yes.
Yeah, good. So today I'm very happy to attend this webinar, and I would like to share some latest progress on our StereoSeq method. Basically, based on this method, we can co-map a whole transcriptome and up to 100 protein markers on the very same tissue section. First, I would like to introduce a little bit about our StereoSeq. So for the StereoSeq, we use this DNB pattern chip. And first, we apply this DNB nanobot on this pattern chip, and we perform the first round sequencing to get the CID. Basically, we know where this spot is, and because of the sequencing. And after that, we generated probes on this DNB nanobot, and here's how these probes look like. So they have poly details, and for capturing the mRNA, and also have the MID for distinguish different transcripts, and also the CID to show to indicate where they are. So after we get the chip, we just apply these sections on the chip, and through our workflow, we will in situ capture the mRNA, and finally we will release this cDNA from the chip, and we will go to the library construction and sequencing. Finally, we will get the spatially resolved transcriptional profile of our tissue section. So there are two main advantages of this StereoSeq, as we can see. One is that it can offer very large field of view. This is because we have very large DNB chips. The maximum size of our DNB chip is 13 by 13 centimeters. That means you can even profile maybe half of the human brain. And another advantage of our StereoSeq is that it possesses this very high resolution. if you further zoom in on these chips, you can observe that the center to center distance between spots is around 500 nanometers. That means if a cell size is around 10 micrometer, then you will have probably 400 spots under the cell. So in that case, you won't get a mixture of gene expression from the single spots. You will get even the subcellular resolution in this sense. So because of these two advantages, we can have very, very high spatial resolution. Here's an example we profiled in the mouse brain. So in the green color here represents the transcript we actually detected in this mouse brain. So we can, even from this image, we can see this transcripts are well separated from each other in cellular shape. And if we merge this DNB transcript we detect and with nuclear staining, we can see these transcripts are really around this nucleus to show that it's really the transcript to get from the cell, from the spatial. And here's another showcase we performed in the mouse embryos. So you further zoom in, and based on this nucleic acid staining, we performed the segmentation, and we did the clustering, and you can really find a different type of cells in the whole transcriptional view. And because of this advantage, and many researchers has already used our SteelSeq to publish the high impact journals such as Cell, Nature, and Science. And one thing to notice that it's a SteelSeq can apply to various species such as mouse embryos and axolotl, and also the human tissues, melanoma cells, and also the Drosophila and the zebrafish. So that based on these publications, we can see the SteelSeq really have very broad applications. So, but we all know a cell's identity or a cell's transcriptome information is only one layer of the information. So to really identify a cell's molecular identities, we need to combine different modalities together. For example, the genomic information, and also epigenomic information, also the proteomic information. So once we gather these two, all the modalities together, we can really precisely identify a cell's identity. And that's why we really want to invent kind of this tool to look into this in spatial. And here's the data we generated on generated from the published study. So it's a single cell seq data. So if you only use RNA to cluster these cell types, you maybe find the CD8 and CD4 positive T cells are not well separated. But if you combine protein information together, you can find that cells population are quite well separated. And if you further zoom in this data to see which information is contributing more in this clustering, you can see that protein information contribute more. So in that sense, protein information is really necessary to accurately phenotype cell identities. So that's why we want to develop this SteelSeq method. So we want to simultaneously detect gene expression and also protein expression on the very same tissue section. So here is how does the SteelSeq works. For this version, we use TotalSeq-A antibodies from BioLegend. So here's the structures of this TotalSeq-A antibody. It has the antibody, also has the oligo attached to it. For the oligos, they have adapter for the PCR amplification, and also the 15 NT barcode for indicate which antibody it is. So also it at a 3' end, it has 32 NT poly-A for poly-T to capture. Because of the poly-A tails, we can use our chip. Here's the probe on our chips. It has CID, UMI, and poly-T. We can use poly-T to capture both mRNA and also ADT molecules at the same time. And through kind of our chemistry, we finally will generate mRNA library and ADT library, and we subject to sequencing. We will get both mRNA and also protein profile from it. that's the basic idea of the stereo-cell-seq. So first, I would like to introduce how could we manage the bin size to obtain different spatial resolution. Because of the resolution is quite high and the spot is quite small, it's around 200 nanometers. To get enough counts for the downstream analysis, you need to make it different bins together. For example, bin 1 represents only 1 DNB spots, and bin 2 represent 2, and bin 3 represents 3. So by leveraging this, we can get enough kind of features or gene expression counts for the downstream analysis. So here is actual data we generated from the stereo-cell-seq. So this data were generated from the mouse spleen. So in this enlarged picture, you will see that this resolution is at the bin 10 spatial resolution. So if we compare the existing method generated from 10x Vision platform, we can see the resolution is much higher. Even we put it at bin 10 spatial resolution, you already see these fine structures of the CD4 positive area. And here's another example we performed in mouse thymus. So this time we perform F staining of CD68 positive macrophages, and also perform this stereo-cell-seq on the very same section. So we can see from the immunofluorescence, of course, we can see the single cell resolution of this macrophage are well separated from each other and show very bright signal. And here's the data we generated from our stereo-cell-seq data. We can see even at bin 10 level, you will see this signals are separated from each other, and they are actually we can reach single cell levels on these tissues. And next, we would like to know how is the reproducibility of our current workflow. So we took 3 sections and we performed the stereo-cell-seq on these 3 sections. And here's the data we show for the gene expression. So with the similar sequencing saturation, and we get very similar genes and UMI is from all these 3 consecutive sections. And after that, we performed bulk correlation between different sections. We can see the correlation is quite high. Also, the overlapping genes is a majority. Next, we perform this spatial clustering based on all the bin 50 cubes so get from this stereo-cell-seq data. We can see here's the UMAP, and if we observe this distributions of this bin 50 across the different sections, obviously there's no significant difference between these 3 sections. And after that, we map this cell or cubes to their spatial resolution to the spatial context. We also didn't observe any very big variance across different sections. And the next, we investigate whether the protein expression is also very consistent between these 3 consecutive sections. The first thing we look is that we observe this pattern. So in this each sections, we put around around 15 antibodies, and if we observe this antibodies expression patterns, we can see these patterns are quite similar across different sections. And after that, we perform the bulk correlation analysis, and basically we use all this antibody information and to see whether this section is similar with the adjacent section or not, then the correlation coefficient is quite high. And after that, we also perform that the clustering analysis, also very similar to our gene expression results. We didn't observe any significant variations from the protein expression level. So based on the protein expression and the gene expression level, we can conclude that our stereo-cell-seq workflow is quite consistent and it has good reproducibility. The next thing we wonder that whether our stereostatic is accurate or not. So in this example, we compare our stereostatic expression patterns with the immunofluorescence patterns. So in this case, we use CD8 antibodies to stain the CD8 positive T cells, and we can see, judging from this signal distribution is very similar. Also, we perform this correlation test between the ADT UMI signals and also this IF signals. We can see the correlation is quite high, indicating our stereostatic has very good accuracy. And after that, we also invest the other cell markers such as endothelial cells for CD31 and also macrophages, for example, CD68. They all show very good correlations. So based on this data, we can conclude that our current stereostatic workflow is quite accurate to reflect the true what is happening in these tissues. And the next thing we did is that we used the commercially available cocktails from the TotalSeq-A. So the TotalSeq-A, because it's quite limited, because now they only provide 128 kind of antibody cocktails maximum. So we use that antibody cocktails, and we investigate whether we could co-detect RNA and also this high-plex protein information on the same tissue section. And we compare the stereoseq only group. We can see with the very same similar sequencing correlation. Also, the statistics looks quite similar. And also, we perform this clustering analysis to show there's no significant difference, even we add 119 proteins into this workflow. And here is selected protein markers that we put in this experiment to show that their spatial expression pattern is quite unique from each other. And the next question we would like to know is that even though we could do 100 plus proteins, so whether if we include this or scaling up this plexity will influence the RNA or the protein expression. So in this experiment, in these three consecutive sections, we put 10 antibodies and 30 antibodies and 119 antibodies into the experiment. We first investigated whether we could get similar gene numbers from it. The answer is yes, and also from the clustering analysis, we didn't observe quite a significant difference. And also from the protein level, so we compare this 10-plex versus 30-plex, and to see whether the protein we put in the 10-plex is influenced by adding another 20 antibodies. The answer is no. So that even though we add up the antibody numbers, we still get very high correlation between these two sections. And here's the result. We compared the 10-plex with 100-plex. We got very similar results. So even though we scale up the plexity, it doesn't influence RNA and the protein detection. And the one interesting thing to know is that in our experiments, we also found that we capture much more ADT counts than the mRNA counts. Here's the data from a published paper. They are using 10x Rhythm platform, and they compare this ADT tags and whether they are correlated with this B cell enriched gene. So here on the x-axis, it represents the counts added up together of these three genes, and also on the y-axis, it represents the counts added together based on these four markers. So we can see that from their experiments, the ADT tag expression is much higher. You see the tenfold, nearly tenfold kind of increase compared to the mRNA data. And this also is, we also observe very similar in our data. So on the first row, we select some genes from the stereostatic, and also here in the bottom row, we selected some markers for their respective genes. And we can see the expression level is indeed much higher than the gene expression. And for some genes, you can barely detect it from the transcriptional side, but you can detect it very well from the protein information. And further things I would like to point out that after that, they use this ADT and mRNA to do the integrated analysis, and finally they found that ADT information contributes much more, as we mentioned in the single cell RNA data. So in the spatial context, also ADT information contributes more to distinguish these cell subtypes. So also, that's the reason why we want to compare. Also, that's the reason why we want to add this protein information. So our collaborators from the A-Star, Uma, and from Jonathan Lab now is investigating this data. She was trying to use this integrated data to show whether how could we use this protein information better to maybe we could identify cell subtypes or get some biological insight which we won't get from the only RNA-seq data. But because of time, I won't talk too much here. So at last step to show that other applications in other tissue types based on our stereostatic method. Here we show that our data generated from mouse spleen, we can nicely see this CD4 positive T cells and IgG positive B cells, and also CD169 positive macrophages to form this red puff from the mouse spleen. And also mouse liver data, we can find this positive TR119 positive red blood cells, and also the cellular cells around it. And at last, I would like to show the data we generated from the human lymph node. Also, we can see even though the IgG and also the CD19 represent for the B cell types, we can observe quite different spatial distribution. This indicates that maybe we could use this special information to really distinguish these cell subtypes. So with that, I would like to end my presentation. I would like to thank people who involved in these experiments, and thanks for our collaborators, and thanks for Jamie for the collaborating and coordinates for contact their collaborators, and also of course thanks my teams for generating this data and analyzing this data. Thank you.
Thank you so much, Dr. Heng Yang, for this enlightening talk. We are excited to hear the latest advancement of this stereo-seq technology with stereosite-seq. We can now detect RNA and protein multi-omics simultaneously in the spatial biology technology, right? Is there any question? At this moment, I do not see any. Maybe I'll just throw in one there. So it is quite interesting, right? And I guess it's not just from this that you've shown that the RNA transcript and the protein expression level has some discordant, and it's actually very interesting. That's why multi-omics studies are needed. What do you think is the role for protein degradation in terms of the functions and this, in diseases and development?
I think protein degradation could be very important to investigate. For example, in the Alzheimer's disease, because of the fold of alpha amyloid, maybe this could be kind of contributing factors to the AD pathology. If we could understand how could these proteins degraded, maybe we could think of other kind of treatment to promote these degradations. Maybe the symptoms of these patients could be alleviated. That's my thoughts.
Thank you. Thank you. I think we have another question in the panel. Does each ADT, antibody-derived tag, correspond to mRNA, which coded this protein in the cell? So I guess it's a very similar to my question, right? So it's a do you see sometimes one or the other that's not corresponding to each other?
Yeah, so it really depends. Also, from the bulk level, we can see that sometimes the gene expression level and protein level are not very correlated for some specific target, but for some targets, it's very consistent between protein and also the RNA level, but for some proteins not. And I think that's maybe the future kind of directions to really further kind of look into this discrepancy and to figure out why it acts like this, and maybe we could find some interesting stuff there.
Great, thank you. There's another one. Given the 32 nucleotide poly-A primer for capture, I don't see there's a question. Right. I guess there's also, is there a possibility, I'm trying to interpret this question, and please, Derek, if I'm wrong, just type in more. Since there's a 32 nucleotide poly-A primer for capturing AET, right, is there a possibility that actually you're also still capturing the RNA part?
Yeah, actually, it's not only 32, because I think maybe the audience refers to that the paper from this paper, they use kind of around 32 antibodies to put together to detect these protein expressions. But actually, in our experiments, we use much kind of high complexity. We use around 119 antibodies from the total-seq from the BioLegend. So actually, it's more than 100 plex.
Yeah, I understand that this is amazing. We can actually do 119 protein detection at same time as the RNA transcriptome, right? In this technology. But I guess maybe we go further back. There is a slide that you're showing the molecular design of stereosite-seq.
Yeah.
There's the poly-T, right? 32 nucleotide poly-A. So I wonder whether it's asking that it also captures the mRNA part.
Yes.
In poly-T, right? But then you can differentiate since you have the nucleotide barcode.
Yeah, that's correct. Yeah.
Right.
Because we have this barcode, we can distinguish this kind of the ADT information from the mRNA cDNA information. Yeah.
Okay, great. Derek, I might have misunderstood. If anything, please put in more into the questions. Okay, great. If not, let's stop here. Thank you so much, Dr. Heng. And we'll conclude now. I really want to extend my appreciation with all the speakers. And let me go to the next slide. Right, thank you all for the engaging questions and discussion. We hope this webinar has provided you with valuable insights into our latest tools to investigate immune profiling. As a reminder, all our webinars, including this one, will be available on demand on our platform, and our guest speakers will answer any questions online if not done so yet. We encourage you to revisit the sessions and explore the wealth of knowledge shared on the website of Genomics Unlocked. Just to add another point, actually, except from the BGI research, all these are published app notes as well from Takara, from Singleron, as well as from this 10x conversion, right? And you're welcome to ask, and we will share these app notes with you. Thank you once again to our guest speakers and all participants to make this event a success. We look forward to hosting you in our future webinars. Until then, stay curious and continue exploring the vast realm of immune profiling and multi-omics. Thank you all.